Good afternoon, everyone. I, Jatasya, welcome you all back to the conference. And without any further delay, let's be prepared for another enriching session. It is with great honor that I now introduce Dr. Avinash Mishra, CEO of Grodia Technologies Private Limited, a computational biologist and data analyst. Mr. Mishra is an IIT Delhi graduate specializing in drug discovery. Their innovative approach includes an in silico drug discovery platform utilizing efficient algorithms such as machine learning and AI. Their aim is to accelerate drug discovery processes and improve research quality for experimental biologists. With a strong foundation in statistical modeling and data interpretation, his expertise extends to high-performance computing environments. He and his team are actively engaged in research, clinical data analysis, and healthcare application development, collaborating with clinicians to design powerful tools for disease prediction. Let us come together and offer a warm welcome to Mr. Mishra as we eagerly await his inspiring session. This session will be chaired by Dr. Dinesh Kumar, and I would like to invite him on the stage to honor Dr. Avinash Mishra.
Connection release है नहीं
So good afternoon to all of you. I think all of you are settled down now, and now we can start this session. First of all, thank you for giving me this opportunity to come here to actually talk with you. Okay, so just take this presentation as a conversation between you and me, and less like like a presentation kind of thing, or talking anything really challenging. So the company that I have started when I was in my PhD, and in fact I finished my PhD from IIT Delhi, is called Grodia. So this is the company that I started, and as the name suggests, that the company has two component, growth and idea, and of course growth can happen with idea, and sometimes may not happen, right? So you might have a brilliant idea, but you may not get the growth. Anyways, we think that whatever the tools or the skill set that we have at that point of time, so can we commercialize it? Can we monetize the concept that we have? Okay, and that's why I can see students, definitely more students than faculty here, and that's why I said let's talk about how science can be monetized or can be commercialized, and it should be, right? It's great to do a fundamental science, and that what I also have done initially. So the overall mission and the vision of the company is that how we can reach to the experimentalists, those who are doing some biotech, pharma experiments with our computational algorithm, and say that what you should do and what you should not. Okay, so this is the prime objective or the vision or the mission of the company, that we should reach to the experimentalists and say that okay, whatever you are doing is going to give you some fruitful result or not, and in this way you save a lot of time, lot of effort and money and capital, everything for that particular client, right? So and we do work in small molecule drug discovery, okay, protein design, genomic data analysis, and some of the secondary data research. I will come to that point, and you can ask any question wherever you have a doubt, maybe at the end of the session. Now, as I said, I started this company in 2018, end of 2018. In fact, it was 2019 onwards, and from IIT Delhi, and the whole idea was definitely to show your skill set that it is really useful for something which is going on, right? So our expertise is in silico life science. In silico means computational life science, okay? Data analysis, AI, machine learning application, development of tools, which is in the area of life science. These are the three key elements we work with. Machine learning based discovery, okay? So as we all know, right, that there is a huge impact of machine learning in almost every field, and drug discovery is also one of it, okay? And you know, the reason why we have the machine learning these days with lots of impact is because we have a lot of data. We developed a very sophisticated algorithm now. Now we also have a strong hardware, right? So to handle that data set. So this is the time for the machine learning. Whether one's accept or not, that's a different story. But we have to, you know, we have to appreciate that what it can do. But at the same time, you also have to know that what are the limitations of machine learning, right? Because my background is mostly on the science side. So I use machine learning a lot, but then I also understand what are the limitations when you are dealing with the scientific problem. Then we have in silico simulation like molecular dynamics simulation, protein, ligand, ligand, small molecule. And then we also develop algorithm which can quickly answer, like if you want to know the toxicity of a compound, or if you want to know the solubility of a compound, those things which have discussed in the earlier session. So this is the first segment, and because I will keep my technical talk a bit short, and then maybe you guys will be open to ask question. So the first thing is the compound design. So we say that compound means a small molecule right now, okay? It could be anything, but here I'm referring to a small compound. So what are the things that you can do in a small compound discovery is you can repurpose the existing drugs, okay? So I think all of you are familiar with this term, that whatever the drugs are approved in the market, you can repurpose for your for whatever the disease you are targeting, and see that what is the chances of curing this disease by the existing drug. And the advantage is because it has already been tested for the safety, clinical trial and all, so you need not to repeat it, okay? So that's as advantage. The other one is new chemical entity discovery, which is NCE, which is much more challenging, and you say that I will do something absolutely new, okay? Then you have to go for all the clinical trial, discovery phase, preclinical, everything, right? So from this scratch. Then there is something called fragment combination, that also we do, and where there are small fragments and then they are linked together to make a drug-like compound, and then peptide and organopeptide. So, and all these we are doing computationally, right? So we are a complete computational solution in therapeutic molecule design, okay? So you start when you say that I need to go for this particular disease, okay? Suppose if it is an infectious disease, or if it is a metabolic disease, whatever the disease are. So the next step is, once you identify the disease, the next step is to identify the target that you want to work on. And the target most of the times is a protein, right? Like GPCR we just heard in the first session. So once you identify the target, the next question which you or we all have to answer this, that this target might be responsible or linked with your disease but is it druggable or not, right? Can we design any drug or not? So that is the first question we asked, and we answered it. Definitely when we ask, we have to answer the question. Then we go for library design. What kind of library design a person is looking for? Because we are like providing services to our clients, so it depends on the client that what they are looking for, what kind of library they want to create, right? So these are the kind of the library, and there are many more, but this is just one snapshot of it. And then once you have a library, once you have a compounds, right? You say that you have 100 compounds or 2000 compounds or whatever. Now we talk about the millions of compounds, right? Nowadays we don't talk about 100 compounds or 500 compounds. Then we are just making a joke, right? Now we talk about million compound, 10 million compounds. Talk about the entire chemical space, and people are doing it, right? It is not like if we don't know, it is not happening. It is happening. People are screening million compound, and then, but then your method should be very fast, right? Your method should be very fast. So these are the methods. Like on the left hand side, on the right hand side, you have the physics-based principle. When you say docking, targeted docking, molecular docking, and all, a blind docking. And then on the left hand side, you have methods that are principled by machine learning, right? Deep neural network or convolution neural network, some scoring function that you can design using the machine learning or data analytic. So if you combine both of them, so the solution is when you combine them, okay? Solution is not machine learning alone. Solution is not physics-based principle alone. Okay, so and then there is something called molecular dynamic simulation, where you put your protein ligand and see the different kind of motion that it can perform, and we have a set of portfolio for molecular dynamic simulation where we go for protein structural variability or membrane protein, enhanced sampling. So all these methods are, I'm not going deep into the method, but if you have any question at the end, you can ask that, okay, what is that, what is that? But what I can say is all these methods are in sense of when you perform molecular docking or any AI ML method, you need to know whether it is really true when the system is flexible. So that is answered by molecular dynamic simulation, because your biological system is always in motion, so you cannot answer by rigid method. So I said, drug discovery was going fine. Computational drug discovery was really going good at not a very high speed, but and the COVID is a major factor. Recently, in the COVID, you would find a lot of publication in that domain to answering some therapeutic solution. And then you also have now experimental data, lot of experimental data being generated, which can be used in machine learning to answer some of the vital question that you can see on the left hand side, like virtual screening or ADMET, synthesis prediction, right? So you can predict that this compound has it can be synthesized in the experiment or not. That can also happen. You can also produce or generate new compounds. So this is really very exciting. You can generate new compound, de novo molecule generation, and then AI based chemical properties, which is quite old, because earlier also it was there using linear regression. Now it is being answered more accurately by machine learning. This is the first protocol or the pipeline that we designed. So we designed for initially for one client, and then so it is saying from target to the molecule using the physics-based principle. So this was the first in silico a pipeline. Then we also started working on the AI-based prediction, like what will be the IC50 of this compound for the particular protein target. Okay, so that is what we have designed using graph neural network. And Then there is another classification method that we designed. It said that if there are 100 of or 1000 of molecules, can we identify the true inhibitor, and can we just classify them and say that these are the true inhibitors and these are the decoys. Decoys are like they look similar but they are actually not, right? So this is the AI based method. If we use this classical physics based principle for such kind of problem, it will take huge amount. And don't think about the experiment. You cannot do experiment on million compounds. But if you have this machine learning approach of classifying it, it can be done in a maybe in few hours. Okay. So this is just a small detail. So the good thing is, there are people who are who are having expertise in machine learning because they are from the computational background. But when you are trying to solve these kind of problems, like which is a pure scientific problem, it is always good that if you understand the science. No, one of the principle I believe in machine learning is you get what you feed. Okay, so what you are feeding is very important. So, and what should be fed can only be answered by the person who knows that if this will be fed to the model, what will happen, right? So scientific understanding is required to solve such problem even using the machine learning. So you can see that on this side, I said that what are the components that I'm going to extract. Okay, now you could have your own components. I had my own components, and then those components will be fed to GCN and the other layers of your neural network architecture, and then finally it will be predicted. Okay, and similarly this is also for the classification. This is for the regression, and this is for the classification model. So classification is saying good, bad. Regression is saying how good, how bad. And then this is the time when you assemble everything. Okay, so this is just a one example. We have assembled, make the pipeline very comprehensive by assembling all the components that we have built in this period. So we said use your machine learning, use your physics based model, use the machine learning again to come up with really good at the end. Okay, and then why not use your machine learning to generate new compounds. So okay, with a particular property, you say that I need a compound which will have such and such properties. So that can also be answered by the machine learning component or machine learning algorithm. It's like very much like you generate a new image, right? You might have seen that this picture is AI generated. There is no such person actually exists. Okay, so you can also design a new compounds. Okay, so these are the case studies we worked on. We worked on toxicity prediction, and these are few case studies. We worked on a neuronal disease, ALS, where we designed compound and the compound were tested in the experiment. Every time whenever we design something, it was tested in the experiment. So it is not like that we are just talking theoretically. So not by us, but by the client. They took it and then they tested it and then they found the result. Okay, there is one package in the market which is called Schrodinger. I just had a talk right now with TPU, and he was also mentioning about this software. So he asked me the question that how your things are better than Schrodinger. Okay, so this is not a new question for me because I have been asked for this question many times. So what I exactly told him is when you are trying to do some scientific stuff like this, designing a compound or coming up with some therapeutic molecule, it cannot be answered by a package, believe me. So it is more like a solution rather than a software. Okay. So and every scientific problem may have a different approach, slightly different approach to solve it. So that's why. But yes, if somebody is very rigid, that no, I don't believe all these things, give me the numbers. So we also have the numbers. We compared it with the Schrodinger and we found that yes, it is working better than the Schrodinger. Some of the protocols that we designed, which overlap with Schrodinger. This is one of the case study I did with IIT Delhi, where we targeted SARS-CoV-2 and we said not just one compound, we can hit the protein by 2 compounds. Okay, and then we tested in the experiment and we found the result in the experiment also. This is the team that we have. Right now we are almost like 10 people in-house. We started from IIT Delhi and now we moved out from IIT Delhi. And now we are located in a place called Gurgaon, which is close to Delhi. We all know maybe. Yeah, so that's it. So if now you have any question, any query on anything that related with, because lots of people are, I can see students. If you have any question on the startup or on the science side that we are doing, you can go ahead and ask your question. Yes.
Thank you, sir, for amazing session. Sir, I have a question that you said you started in 2018. तो सर ऐसा बोला जाता है कि जो ऊपर बैठी बड़ी मछली है वो छोटी मछली को दबा देती है. So is it true or not?
Yes, it is true.
तो सर आपने इसे कैसे कोप अप किया?
क्या बोला आपने कि मछली
ऐसा बोला जाता है जो ऊपर बड़े लोग बैठे हैं.
So big fish eat small fishes, right? बड़ी मछली छोटी मछलियों को खा जाती है. But the answer of this question is कि उतना छोटा बनने में भी टाइम लगता है कि आपको खाया जा सके, right? So in business it is not bad that you have been eaten by someone else. So the bad is if you are not paid for that thing. Okay, so if you are well paid for being eaten, so it's okay to be being eaten, right? So but how you get the attention from the big pharma is not an easy job. Okay, so yes, of course, if you see the market, always the drugs are coming from few companies. Does it mean that none of us are doing any research? Yes, of course we are doing it. Lot of academics are doing a research, but then they also understand that they may not have the capacity to take it to the last level, है ना? हम सब कुछ नहीं कर सकते है ना? So either you collaborate or you just outsource your technology. So there is nothing wrong in that. Okay. So in US, what the new students or the graduates are doing is they come up with company. They make a good value of the company and then they sell the company to a bigger one, and then they start a new one once again. So there is nothing wrong. If your objective is to develop a technology, you are achieving it. If your objective is to make money, that's also you are achieving it.
Thank you, sir.
Thank you for very recent answer. I have questions regarding your ligand protein interaction. And you said that you have also been identifying some of the ligands. Now, suppose you have a protein kinase, which is enzyme, and which actually can be active or inactive conformation. So when you look for compounds, will you target a particular conformation, or you will just do a blind scan without any additional information, purely physics-based information, which is the topic?
So yes, we do. So the one line answer is yes, we do, and of course we should do, right? So when we say that this particular protein can exist in active and inactive form, so so the very first step is to understand the conformational variation when it is in active and when it is in inactive. So the first thing that we should do is take the structure of a protein in apo form without ligand and run a long simulation, and then start sampling the binding site or whatever the site you are interested in, and then see that what are the conformational changes that you are observing, and then which conformational change is most stable, means what is the conformation which is most probable during the cycle, that we should hit. So if suppose lots of people might be doing the docking and they start with the crystal structure, so now we also need to understand or appreciate that the crystal structure is crystallized form. Is it the 100% native or not? So that is always a question, but of course it is very close to the native. So we have to take it, but it will be good even in those proteins that are not conformationally variable, like active and inactive. In that case also, I would suggest that if there is a huge or good compute resources, do a simulation for a apo form of even a crystal structure, and see that what is the conformation which is most probable during the 500 nanosecond simulation, and then take that one as a binding pocket to screen your molecules.
Second one, in continuation, do you think that adding some experimental data, for example, NMR may not give you immediately the structure, but you can provide you information on binding epitope. Would that help you on your software in guiding it and doing it in a short period of time? Have you tried that model as well?
So we do use experimental data, of course. I mean, all the machine learning models that I talked about, these are trained on the experimental data only. If we have the experimental data like what you are saying, the binding epitope, even all kind of experimental data is valuable. It is like how you want to use it. Like if I suppose took 10,000 protein and ligand structure from the experiment, let's say protein data bank, PDB, experimental, and their corresponding IC50, and then try to understand that what kind of contacts are necessary for a native complex formation. So these are the experimental details, and then you trained it, right? And then you train your machine learning model on that. So suppose now any software is giving you any complex, you check it on your machine learning model. Is that validated by your principle, like this kind of hydrogen bonding, that kind of hydrophobic contacts? So yes, so if we have any experimental data, only the thing is that you should need in a certain amount. I mean, maybe the very less number of data points might not be very useful in machine learning approach. It might be good in empirical scoring function kind of approach, but not in machine learning.
One last before and one my to other. Have you ever validated your software? Sorry, your
Solutions.
Yeah, results. For example, there are a lot of known drugs which are already in the market. Now you take your native protein and then screen.
Oh yes, yes.
And then it comes out with a solution which is actually in the market. Does that happen?
Yes. So this slide is actually saying that if you have 10,000 compounds and then there are 10 compounds that are already known, some of them as a drug, some of them as a failed drug, but they showed some activity, right? And you mix them with 10,000, then you pick it up using your machine learning or your algorithm. So I'm saying that I can capture them in top 35 100% of the time in most of the cases. So every line is a protein target. So if top 35 I am picking it up, your compound is there. So that's what I did. Okay? Okay, one question.
Sir, my question is, what can be the major reason that India has such a small number of just startups or such a small number of commercialized research as compared to other countries like US or?
तो बहुत ही philosophical question है. It's a mindset. So, see, if there is something large like India, so it will take time to make a change. That is the limitation of being big, right? Like for us, we are a 10 people startup. You tell me tomorrow ki there is one thing which is coming in the market and it has a lots of commercial value. Can you work on that? I may work, right? But you go and talk with Accenture and say that will you work on this? They will not change their protocol, right? Because they are big. So we are big, right? So as you said, India is big. So any change will take time and mindset takes extra time, right? So I mean उसको change करने में तो बहुत time लगता है. But what I have realized, like in this space, 2018 I started this company, but I am in startup from very first year of my PhD. मतलब मुझे उसका थोड़ा I had a inclination towards the running a startup, understanding the ecosystem and all. So what I realized, and then I attended lots of contests, prize और वो जो भी थे ना competitions. And then I came to know that in last 10 years the mindset is changing, ठीक है? But to reach to the last level or to the academic level, it will take time. And this is someone like you have to say कि I will take the risk and do it, okay? So if you decided, you will do it. So like in my case, when I was doing PhD, so I was also thinking about what next I will do in my startup. So it was challenging, right? It was difficult, but that what excites me. That what like I like doing it. So if that is the case, you can do it. But if you're expecting it on a larger mass, it will take time. But it is happening. It is 100% happening in India. Yeah.
Thank you, sir, for a wonderful session. My question is like we perform some pre-formulation parameters to check the interaction between polymer or any drug moiety to have like greater solubility and solubility studies for any kind of formulation. Can machine learning or AI be the alternative for these kinds of conventional pre-formulation studies or not?
So it's a really good question because these kind of compute intensive job, like which is answered by, if I understood your question correctly, it is on the formulation side, and you want to understand the polymer interaction with your certain compounds in the formulation. So in fact I'm working on this. So it is a bit difficult because these are multi-molecular interaction, right? It's even in molecular dynamic simulation, you just put sometimes just one molecule of your target, one molecule of your compound. But in actual scenario, it is not. There are many molecules you use with the concentration, multi-molecule system. So, but right now there is no direct solution. But one thing that I'm thinking about, and whenever I get the chance to say it, I say it. I don't keep it confidential. If someone wants to work on it, that can molecular dynamic simulation be replaced exactly by the machine learning? So in molecular dynamic simulation, you are getting a different conformation at a different time. Can we predict those conformation? Rather than doing it, can we just predict it? If we know the initial conformation, can we predict all the later on conformation? If that is so, then, and there are some initial papers on it, that how machine learning can answer some of the question in the molecular dynamic simulations.
As in software, like for studies can be conducted. Like they claim that if you put one molecule and you can get your solubility studies. So can we go further with that studies or not?
सॉरी यार मैं सवाल ही नहीं समझ पाया.
Sir, like there is
Mastro is one application you are using.
yes, sir, viral Schrodinger.
Schrodinger.
Yeah, so they claim that you can perform some kind of solubility studies at your initial phase for the drug polymer interactions.
अच्छा, okay.
Can we this data is relevant to taking our studies on a further perspective?
I mean, it depends on your research problem. In your research objective, is the solubility is one of the key factor or not? Like for every drug, of course it is. So if I'm going for a small molecule therapeutic design, if I have the solubility data, it will be useful, okay? And because everything mostly is happening in a water solvent media, so I would say yes, it is important that your compound is soluble or not. Yes. Okay. Okay, then thank you everyone. Should I'll take this.
Thank you, sir, for this wonderful and insightful session. And I hope that our audience has definitely taken something valuable from this session. Now I would like to invite Dr. Ashish Kumar Agrawal on the stage to felicitate Dr. Mishra with a token of appreciation.
अच्छा कर उस तरफ सर आई सेंटर इधर ओके परफेक्ट थैंक यू यू वांट टू इंट्रोड्यूस मी प्लीज
It is our privilege to welcome Dr. Prakash Muthudos, a distinguished leader in techno-commercial expertise, to our esteemed gathering. With an extensive background in techno-commercial leadership, Dr. Prakash brings a wealth of expertise to our event. His proficiency spans material science, formulation chemistry, and a solid-state characterization, coupled with a deep understanding of quality by design and process analytical technology. Dr. Prakash is adept to Lean Six Sigma methodologies, employing them for root cause analysis and project planning. His skill extend to statistics, machine learning, and data analytics, enabling scientific insights and strategic decision making. Notably, Dr. Prakash has made significant contribution to pharmaceutical analytics, healthcare analytics, and product strategy. We eagerly anticipate his insights, which promise to enrich our understanding of diverse domains ranging from pharmaceuticals to AI/ML organization. Please join me in extending a warm welcome to Dr. Prakash. This session will be chaired by Dr. Ashish Agrawal. I would like to invite Dr. Ashish Agrawal on stage to honor Dr. Prakash.
Where does this slide go? Okay, I'll have a mic. Hello.
Hello.
Yeah.
Thank you. Thank you for your wonderful introduction. And I start with thanking EHU, faculty, HOD, students for hospitality and a very nice interaction during these 2 days. Okay. And that's a great opportunity. And this is my first visit to Varanasi, and it's a privilege and a kind of a privilege trip I'm doing it. The talk that I will be presenting to you all will be is from plan to build with a touch of data science, Lean Six Sigma. Let's see how it goes. First, let's get into the topic, right? I enjoyed these 2 days, and thanks for the friends and the guys who are interacting right from the receiving us from the airport to the hostel and the guest house, and then on, and the last one, right? And today morning, thanks G for the pile on things. Yeah, very nice. Excited with that. And with this, let's get into the sweetest of the topic. Yeah, and this work that I will be presenting to you is as part of my retainer consultancy with the Neoperk Agrotech company, where they build the sensors for the farmers and try to understand what kind of fertilizer should be suggested using near-infrared spectroscopy and the data science of it. And the agenda, I try to keep it simple, but the slide number will be slightly higher. There is an acronym MAD approach with some examples and a summary. Yesterday the guys were discussing with us regarding the career and how we grow over the things, what should be chosen and all those kind of things. Just to give them what we are here, who are presented since yesterday, you are just seeing the tip of the iceberg. But a lot of efforts have gone into that, and you can systematically do that, and you can design your career, how you strategize from moving from positions and dwelling into the knowledge. If you stop learning, you are stopping your growth. Yeah, and because of this graduation and certain things that I have done today, I stand as a consultant. Right from I have been working for Sanofi, Dr. Reddy's, AstraZeneca, and I have published 15 plus research articles and 2 publications, and then a patent. Those kind of things, but the major strategy in all these places I have kept was break down the complex problems into simple workable solutions so people understand and support you. Yeah, and recently while moving into the business, I just wanted to have a collaboration not only with technical people, also with the business leaders, because the simple funda is if you are with the group whom you are, you grow in that nature, right? So if you want to grow as an entrepreneur and you want to be in the business side of it, start moving along with the business people. When you want to be more like a scientific person, move along with science domain and the academics and many other people. So, but these things doesn't go in silo. It has to be a collaborative effort, right? And one thing that I learned here was this one. Confidence level and the trust score, we call it as. So people don't believe each and everyone just like that. Okay, they can be friends. Shall I ask you one question? How many of your friends, if you call at the night 12 o'clock, pick a phone and say, I'm there for you? In any of the things. How many of your friends? How many friends you know? So 50, 40? And how many friends will come? 2 or 3? right? Yes. That means you have developed the trust curve with that person, or he has developed the trust curve with this person. Even in the business world, even in the companies where you are going to go and work, students, just try to understand this simple logic. You will build the trust with the person whom you are going to work with. Here, many of the companies who have come here, they have told we always take first time the students or the new people as a intern, right? That doesn't mean we are not believing you. We are believing you, but we need some more time to trust you on your efforts and things. That's the only reason we take as an intern. That's only 6 months. It will just fly up. And you set in the initial 3 years, set the period of learning than earning. That always helps you a lot. Other people will have difference of opinion, but we are happy to debate and disagree, all those things we can do. Yeah? Yeah, so I have just not grown on myself. I would not say it's a self-made career of mine. I would like to thank everyone over here. Since from my school teachers, my college professors, my PhD supervisors, my collaborators, and some of them whom all I have highlighted, they have also accepted to be part of the A to Z 4.0 advisory committee. And one person you will know here is the Professor Dinesh Kumar. And yes, just now let's get into the things. Like A to Z as an acronym as MAD competencies, where M stands for Material Science and Testing, A stands for Applied Data Science, and the D stands for the Decision Science. So we try to understand the concepts of materials in D, and then try to apply the statistics, machine learning, chemometrics concepts in bringing down the data tsunami to a readable, interpretable information, and then we give it to the management or to the clients as a decision making, yes or no, recommendative systems like binary decisions. That's our strength. And I engage with academics 33%, and with corporates as a project-based consultant or as a retailer consultant 67%, and that's the various companies that we have been working in the past 2 years, and we are even in discussion with few more companies, and quite soon we will also crack on that. Yes, the topic that is given to me here is the introduction of all the things. I wanted to go fast. Plant to pill. Most of them would understand in pharma this is a very simple subject called as, not as simple, it's a complex, one of the complex subject called as the pharmacognosy, how the plant-based materials can be converted to a medicine. And this can be plant materials, algae, or say for example microscopic fungi. They can be given as usual like an English medicine, or as a Western medicine like a tablet, solution, or an emulsion kind of a concept. But the most problem that lies with the pharmacognosy products or the Ayurveda products is they are complex mixtures and there are no distinctive active ingredients. What do we mean that is? If you say crocin as the tablet, the distinctive ingredient there is the paracetamol. But if you take any Ayurvedic product, churna or some kind of a tablets, there is no ingredient, we might not tell. Even whatever we are consuming as a food, for example, dal curry or something, that also is a food only, it means medicine in a way. So we don't have a distinct one ingredient, there are many ingredients that composites the thing. So understanding those things is very complex. And this is where, for example, if you see, this is entirely by the US FDA, it's termed as the botanical drug substance and the botanical drug product. And the various things like right from the selection of the plant materials, how they are isolated, what is the physicochemical properties of the materials, these all have to be understood. And there are a lot of techniques that are used. And the US FDA calls this as a single concept called as totality of evidence. What does that mean is, right from the point one, if you see, it should be well-controlled botanical raw material. Say for example, if you pull out neem leaf in the month of March versus in the month of October, there is a lot of variations. The variations here means it's not reproducible or repeatable. For example, say like if you are going to make a tea at home and mom says take a handful of sugar and put it in the tea, the person individual to individual, the palm size will vary. So when mom puts, that will be very equivalent and you will have that different taste. But when we put, either the palm size can be small or bigger. So here that variability, that's called as a variability, and that is removed by just by adding one term called as 1 tablespoon, 1 teaspoon, or 5 mg, 10 mg, 15 mg. So now you can do whatever you want, but that mg only has to go in. So that has to be reduced. This is, we call it as the variability. And then the robust chemistry, manufacturing controls, all those things. All the Ayurveda has been there for a long time, but as per USFDA records, only 2 botanical drug products have been approved till date. One is topical and another one is the solution, which is for the side effects of the HIV, what are the things that come in. So what is that making a big problem, why the botanical drug substances we are not able to make it? It is, say like we have to thoroughly characterize. And if anybody remember yesterday's one of the talk or couple of talks people have been discussing on the biosimilars, and also Dr. Navaratna was discussing on how the complexity of the biosimilar molecules are and what are the problems we face. It's the same problem. You have to thoroughly characterize and we do not know what is there in that, right? So that's the problem. And for the drug substance, that itself is a big issue. For the drug product, there is even more hurdles because they are going to be very, very low quantity of materials that are present in the final product. Say like if you, for example, if you make a decoction of neem leaves, the amount of nimbolite, some ingredients are there which are called as nimbolite, nimbine, and all those things. When those are going to come out, we do not have any clue how much quantity is going to come out. Okay, that's always a problem. This has to be normalized or put in place. Right. I just wanted you all to see in this particular thing because most of the times we can, whatever we have been discussing as a problem, we can just split it in a kind of a root cause analysis and take a small example as a tree and see on the top, whatever you are seeing is just a symptom. We do not, it just appears, for example. Okay, for example, say for example, when we go out in the summer, our skin becomes sunburn or something. The actual problem is we might not have a lot of fats or the skin is not hydrated. That might be the root cause. How are we going to drill down to that root cause? That's where you see we split into symptoms, problems and the root cause. If you go by this way, if a problem has occurred and if you drill down what would have happened, that becomes the root cause. But for pharma people there is another option. If you go the reverse, it becomes the quality by design. If you want to solve problems or if you want to stop problems to happen, that's in the pharmaceutical people's hand. You can go by root cause analysis or by quality by design. One of the examples is, say for example, let's take the example of the botanical drug product itself, which is going to be coming from the plant. If you see here, it can be having a problems on the biological assay, content uniformity as the symptoms. The problem can be that the raw materials, inconsistency in the plant chemicals. Actual problem in the root cause can be the nutrition deficiency of the plant that can trigger some other chemical moiety to be manufactured by the plant than the other one. This could be the actual root cause. And here, in order to understand these kind of problems, we might not have a simple solutions in place or simple mathematical median, mean or a standard deviation or even say kind of a principal component can solve our problems. So we have to get down to the things of data analysis, machine learning, artificial intelligence and machine learning. And I just kept this slide as a cooler slide saying that how the industries are progressing. We are now in the industry 4.0 revolution. AI ML is a must. And one thing I'll say here, and I'll just close it saying in this slide, I'll close it saying that if we are not going to gear up for the AI/ML, and already China is very fast and they are in the way ahead, and if we are not going to gear up to this, we will simply call it as a Nokia effect. We will know only after 2-3 years where we are left behind. And my talk here mostly will now focus on selection of the plants. So we want to drill down to the thing where all the synthesis of the plants can be monitored and can be matured or controlled. That's where my talk will be there. And this is the pretty good slide where you can start seeing the critical material attribute, critical process parameters. How this can be controlled is bring in phytochemical yield consistency, because for the plants we cannot control it after we pluck the leaves. That is not possible. That we have been trying to do it, we are not able to achieve that. So what we decided is, why not, if you have 10 plants, all the 10 plants should have a consistent production of the ingredients. So can we have a strategy to monitor the plants, and can we make that possible? Then we decided, yes, we are now working with the agricultural firm and we are trying to do that and help farmers achieve that. That's where we are trying to understand what is the plants that they are working with, what are the chemicals that are present in that soil or present in that leaves or bark or whatever it is, and then use the near infrared sensor, try to capture the data, then after collecting the data, do the data quality metrics, and then do the applied data science. Finally, what we wanted to give is the decision making for the farmers, saying that they should use the current fertilizer or a different fertilizer, or how much quantity of. What this helps us in precision fertilizer for the plants, like precision medicine for the humans. We are trying to bring in a concept called as the precision fertilizer for the plants, so that the production can be consistently made. So in breaking down, we take the near infrared spectra and then validate it using the atomic absorption spectra, because NIR spectra is always a secondary technique. I'm not getting deep into why that is. And you use the primary technique as the absorption spectra, because we are trying to monitor the micronutrients and the macronutrients from the soil to the plant, which are sodium, potassium, calcium, nitrogen, carbon, all those kind of stuff that comes from the soil. So once we make sure what is coming from the soil to the plant is made sure that is made available, the plant will give you what you want. That's our simple concept. And here the machine learning, we have been speaking about machine learning and there have been many people who are familiar with it, but what we wanted to start with the information here is, just think machine learning as a kit. When we start explaining to the kid, we start with say like whatever red color is apple. This is the data that is given by the parents to the child. Think in the child. Don't think like a grown-up child. Think like a growing child. So when now you say whenever the child sees anything that is red in color, it's going to tell that's an apple. Whose problem is this? Whose problem is this? Why the child is saying everything red is apple? Anyone want to go?
Lack of data point.
Yes? Obviously.
This is the only thing
You are
we
right.
got.
Sorry?
This is the only information taught.
Perfect. Another. Any more? Exactly. Those two things can be taken as an example. If you are not going to give enough data points or these representative data points for that, so what all can be red in color, we have to mention to the child. Then only the child will tomorrow will say if it sees cherry, it will be able to predict that is cherry. Okay, but the things that have started now growing in the same world is we don't keep learning the vegetables each and every time. Correct? Once you have been taught in the secondary schooling or tertiary schooling, you will be now then going to seek your own information. That's where the advancements in the models like transfer learning and those kind of concepts have come, but let's not dig deep into it. And then The problem with the traditional machine learning concepts is it's a kind of a pattern recognition things or trend. It learns the data by knowing the trend. If it is a red in color, it is like this. But what we wanted to also get into in near future, or people have been moving forward, is an artificial neural network where the equations or the physics behind whatever is given to the data or given through the data is understood, and that's where we are starting to go. Why this helps us is whenever you build a calibration curve, we always build the calibration curve from 50% to 150%. But if your new sample comes which is 160%, we won't be able to predict. That's where every model fails. But with the artificial neural networks and things, it can 50 to 150, whatever the equation, whether be y equal to mx plus c or the polynomial equation, it learns by itself. It can also predict 160, 170, 180. Yeah, that we have been validating it, and we are already very confident on those things. That's why people wanted to quickly move from the traditional machine learning concepts to the artificial neural networks, and they have been moving already into the next advancements like graphical neural networks and things. So the problem in that was given to us when the company approached us was they were able to classify the data, good data, bad data, middle data, whatever it is, they were able to easily classify it. But they had a problem when they wanted to do a quantitative analysis of, say for example, how much actually is the sodium content, how much actually is the potassium content in the soil, so that they can change the fertilizer. The main thing of this work is when you are going to change the amount of fertilizer, if you are going to reduce the amount of nitrogen in the soil, nitrogen is at least 20 times more carbon emitting than the carbons. So that's one of the reasons why the nitrogen in the fertilizer have to be reduced. And this is the action point given by the government of India to all the fertilizer companies. And that's where we are targeting that one. Yeah, and this is the thing. And we started doing the, we devised our own plans, and we have our things like data volumetrics, is the model interpretable, explainable, generalizable, and transferable. These are our major concepts. Again, here also we did the root cause analysis, and whenever we build the model first, we always try to answer our first question, symptoms. Is the model explainable, interpretable, generalizable, or transferable? As of now, we have done our model to be the first three, and in future we are planning to do it as a model transferability. That's nothing to do with our sample size or number of samples. What we have identified is the bias in the samples. That bias we are trying to address it. What is that bias is the near infrared spectra that is collected in a plant that is near to the water has a different spectra than the spectrum that is collected far away from the pond, wherever is the pond, that has a different water content, because the movement of water and the carrying of the nutrients are very different. This is a bias. It's not a number of data point problem. It's not a variance problem. It is a bias problem. And then we devise this strategy where we need to understand the data, then statistics definitely has to be included into the thing, machine learning, first principles, where here the first principles is the understanding the near infrared technology, Beer-Lambert's law, and those kind of concepts. And we have published 3 papers on this near infrared itself, and 2 book chapters. And we are already working on a couple of more papers that will be published quite soon in this year starting. And that's the Petio spectroscopy, wherein we take the near infrared spectra, and just to make that, it's just a umpy things, and we cannot easily conclude what it is there. We just do it as a first principle. We just did a subtraction of one spectra with the another, and we started seeing in the regions. We started seeing the regions around 1850 to 2100. Here we try to, from a domain perspective, what we know is this is a water band. So now if the water in the soil is getting transferred through the branch to the leaf, that's where the ingredient transportation is going to happen. It's as simple as that. If the water is not there, there is no ingredient going to get transferred to the thing. Because water is the transporter of all the micro and the macro ingredients. So if you maintain the water level in the soil correctly and equivalently along all the 100 trees or 200 trees, this is going to be excellent. That's the first concept that we try to understand. And this I'm just keeping as a kind of data that is falling between lower to the higher level, and what classification they were able to achieve a better results, because you see something below, something middle, something above. This they can achieve. Classification model performs better, but the problem comes with the regression model. For the regression model, how the data should be is, it should be linear. If you are aware of regression problems, the data always is at the 45 degrees from the zero, and this data now has to be converted to this format. Then only we can do a better regression of the models. So here number of data points is not the problem. Here the major problem is the data is not in that format. The pre-processing of the models or pre-processing of the data now should take care and bring the data to this time. Then your model will perform better. This is where what we did, a simple strategy is binning of the data. So say for example, what is this binning? I will just give it in a simple language. If you have a age group from 18 years to 16 years, and in this election time they wanted to know the youngsters whom they are going to vote, the middle age people whom they are going to vote, the elderly people whom they are going to vote, we cannot run 18 to 60 data points in a single go. There will be confusing, because between 18 and 19 there will be a data point, between 90 and 20 there will be a data point problem. For this, what you can do is split the data into Gen X, Gen Y, Gen Z, or even kind of another bucket, and then start doing your analysis. Probably that will give you within the error you get a better results. That's what we simply did, and we were able to convert the entire data into a regression problem, and the model was performing much better, 0.99. Now we call this as a biased binning method. We are in the process of converting that into automated one, and we are quite there. And what I have shown now here is always try to address the root than working on the symptoms. Be it in any level, be it in education, be it in your career, be it in an office problem, be it a clientele problem or anything, work at the root level, not at the symptom level. Then you will find the solutions quite fast. And then I've shown you how innovatively you can just change a little tweaking of the pre-processing models and we can achieve the results. And the future work that we are working on is how we are going to transfer when a new data is going to come in, how we will convert that. But the model was built in Mumbai, farm soil. We want to take that to Karnataka, Tamil Nadu, anywhere in India or anywhere in the world. That's our ambition. And then, yeah, all those things. And the take-home message is holistic farming practices. Try to address this will, because India is at a very nice level, at the agriculture level. We can help the farmers very well, and then optimize the nutrient management. And then our major target of doing all these activities is reduce the carbon emissions by 2030. That's what we are trying to use kind of optimal usage of the fertilizers. And thank you. Open for questions.
हेलो सर, मैं सेल्फ अभिनंदन फ्रॉम फर्स्ट ईयर आईआईटी बेंगलुरु। आपने डीएसएनडीपी के बारे में बताया, तो एज कम्पेयर टू न्यू जेनेटिक टेक्निक सीएसआर एसपीआर टू वर्सेस डीएसडीपी, तो जब हम डीएसएनडीपी जैसे टेक्निक यूज करते तो रेयर बहुत ज़्यादा चांसेस होते कि म्यूटेशन इंस्टेबिलिटी हो सकते। तो इन टर्म्स ऑफ प्रैक्टिकल एप्लीकेशन इसके लिमिटेशंस क्या है, इंडस्ट्री लेवल पे हम इसे ओवरकम कैसे कर सकते हैं?
I'm sorry, I should have said what is DS and DB. I'm sorry, that's my mistake. DS here is called as drug substance and DP is here, it's called a drug product. Drug substance is nothing but a chemical. DP is called a product. I'm sorry for the confusion. Thank you.
Nice presentation.
Thank you.
I have a very simple question about the supervised and unsupervised learning.
Okay.
Like when we talk about the machine learning, what difference we see with these two learning? Is the unsupervised learning is a true machine learning or what's the real step?
Okay. In the unsupervised learning, it's something like kind of you are adding, say, like you are given a kind of red, green. Say for example, in this if you take, it is sort them, right? Pink, red, yellow, like that. That becomes unsupervised learning. Here, in that either the data will be bucketed or be clustered in a kind of a thing, but it will be only on the data it will do. It will not do on the target. If the target is getting wrong, meaning what is your say? Sometimes you are speaking about the IC50 and you are speaking about the nutrient content, and based on that the features have to work. Then it becomes supervised learning. If you know something and you are working, that becomes supervised learning. You do not know anything. However, the data can get bucketed, or it can get clustered, or it can get like PC and KNN. These are all unsupervised learning. Yeah, but we cannot predict what will happen. Yeah.
Thank you for the wonderful presentation. My question is regarding the last thing you mentioned, probably that is what I wanted to ask you. When you want to make it a bigger, so you have understood one phase, this is the requirement of the soil, you want to transmit that information to some other phase, which you said across or whatever. So what is the roadmap you are thinking about? How do you want to create that?
Yeah, the roadmap that we are thinking about now is the near infrared technique, right? You will get affected because of the particle size, because of the moisture content. In principle, near infrared gives absorption based information, scattering based information. Absorption gives you chemical information, scattering will give you physical information. Now we wanted to develop a model where it will act like a swan. It will only pick up the absorption information, not the scattering. Once that is done right, you can easily transfer it to anywhere in the world, because your soils, it doesn't matter what is the content it's going to do.
Essentially, you will feed the soil exactly the.
No, we are trying to only work on a kind of pre-processing approaches which are not going to affect us because of the scattering information. And we are working in a reverse way. So where I showed you binning, right? When you bin the data, and if you automate that, it will not get affected because of your scattering.
But what about other environmental factors? Would they also impact?
No, that's where I forgot to add one slide here. When you have the absorption information is getting picked up, it's something like, I'll give you an example like human and a swan, or human and a eagle, or standing near the river bank or something, river. A fish is there which has to be caught by two people, right? Eagle can catch it. Human will have difficulty because human has distractions. He cannot see the fish in the water because of the light scattering or whatever the thing, shadows or whatever it is. But the eagle can capture the fish very quickly. We are looking for models and approaches where it's going to be like an eagle, not a human. And we have been finding successful with pharma things. Now we are going to work with, in the pharma we have developed the models where we build on the physical mixture, and the model can predict even the production scale samples. So we have achieved that in the pharma side of it, and we have those 3 papers that I shared, right? That those are the testimony for that work. This one we are doing it for the agricultural areas. Yes, we are there, but it's taking us little time.
One last question.
Yeah.
Everybody wants to leave now. What percentage of drugs right now in the market are plantables? Do you have any number?
Actually, there are many drugs that are planned. Say, for example, one drug, chiconicide, we have. Then you have this paclitaxel itself. Then you have this importantly this that's given for the eye tech that for the muscle relaxants from the Sanofi. There is one molecule that I keep forward. It's a Zinodol tablet. That's kind of a plant. But these are all plant-based. Bromexin, Ambroxol, all these are all plant-based things. These are all part of it, but not the entire ingredient there. Right now, but the botanical is you extract as it is and then you take it. If you take that as a chemical compound outside, you can still, it is specific. But if you take the plant-based material as an extract, that is not exact thing. There can be n number of molecules, there can be 100, there can be 200. We have no clue. So what the regulatory agency says is, you at least tell me what are the first 4 ingredients that are present and what is that for giving the effect form. Design the strategy that way. That's how they say. Yeah? Did I answer your question?
Yeah. Thank you.
Thank you, sir, for sharing your wisdom with us. We are truly grateful for your invaluable insights. Now, I would like to invite Dr. A.N. Sahu to felicitate Dr. Prakash.
Thank you for staying till now.
So I will take. Now I would like to call Hinal and Jatasya to take over the stage. Priya and Jatasya to take over the stage.
इसका जो मेन पेज है ना अपना
Yes, sir. ये सर को दे दो। ये वाला लगाना है।
नहीं 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 माइक है, ये वाला लगाना नहीं है।
अरे
ठीक है कोई नहीं, नहीं है तो
ये लैपटॉप निकाल।
निकाल दो। स्टार्ट करो।
मेन पेज लगाना है।
मेन पे लगाना। तो वहीं से लगा देना। वहीं से लगा देना। इधर फाइल से लगा देना। वेट करना नहीं, वेट करना। Why are they not doing like this?
As we approach the end of this remarkable conclave, I, Priyadarshini,
And I, Jatasya,
are filled with profound sense of gratitude and inspiration. The International Conference on Pharmaceutical Innovations and Spirit, the annual Techno-Pharma Conclave, has been a testament to the relentless pursuit of knowledge and the spirit of innovation. We have witnessed an exchange of groundbreaking ideas and celebrated the achievements that have pushed the boundaries of pharmaceutical sciences. It is now time to honor those who have made significant contributions to our community and the field at large. Our sincere thanks goes to all the distinguished guests whose presence has bestowed an air of distinction upon today's proceedings. Your collective wisdom has catalyzed an invaluable exchange of insights, knowledge, and forward thinking that propel our industry to new heights. Our heartfelt appreciation extends to our sponsors and partners, whose support has been instrumental in making this event successful. We thank Sir Boltzmann Labs and Firmus Vision for their unwavering support. We now turn our attention to the esteemed faculties and principals whose dedication and pursuit of excellence in education and research have not only molded the minds of the next generation, but have also pushed the boundaries of pharmaceutical engineering and technology. Starting with the felicitation of the principals, I would like to request Vinod Tiwari sir to felicitate Dr. Dinesh Kumar of IIT College of Pharmacy, Jaunpur. Now, I would like to request Dr. Shreyans Chen to felicitate Dr. Vinod Kumar Tiwari of Dr. Mahendra Kumar Chote Lal Ben College of Pharmacy, Prayagraj.
Felicitation by
Now, I would like to request Dr. A.N. Sahu to felicitate Dr. Shailendra Bahadur Singh of Shiv Murat Pharmacy College. Thank you, sir. Now I'd like to request Dr. Dinesh Kumar to felicitate Dr. Priya Jain of Lakshmi Narayan College of Pharmacy. Now I'd like to invite Dr. Ashish Agarwal to felicitate Dr. Hari Pratap Singh of Vidyavati Devi College of Pharmacy, Salimpur. Coming with the felicitation of faculties, I would like to invite Shweta Patel of Maharana Pratap College of Pharmacy, Kanpur, Manu Dwivedi of Maharana Pratap College of Pharmacy, Mr. Alok Kumar Mishra of Rajkumar Goyal Institute of Pharmacy, Ghaziabad, Sagarika Kabra of Rajkumar Goyal Institute of Technology, Pharmacy, and Devin Kumar.
वन बाई वन लिस्ट निकालना है, वो लोग आएंगे ना स्टेज पे, एक एक करके आएंगे।
सर एक्चुअली फाइव लेके करें सर।
ओके
हाँ, क्योंकि 43
पाँच का नहीं,
5 का नहीं।
एक बोल के वेट, एक बोल के थोड़ा सा, ठीक है।
I'd like to request all this faculty to come to the dais, please. Okay, starting with Shweta Patel of Maharana Pratap College of Pharmacy, Kanpur. Manu Dwivedi, again from Maharana Pratap College of Pharmacy, Kanpur. Mr. Alok Kumar Mishra of Rajkumar Goyal Institute of Pharmacy, Ghaziabad.
Next.
Sagarika Kabra, again from Rajkumar Goyal Institute of Pharmacy, Ghaziabad. Devendra Kumar Dubey of Institute of Pharmacy, HCPG College, Varanasi. Akash Maurya of Shri Harish Chandra PG College, Varanasi. Ankita Kumari of Shri Harish Chandra PG College, Varanasi. Jyoti Singh of Anand College of Pharmacy, Agra. Sulakshana Pal Singh of Anand College of Pharmacy, Shata University, Agra. Ms. Gunjan Sharma of Anand College of Pharmacy, Agra. Dr. Ashutosh Mishra of A&D College of Pharmacy, Raj Kishore of Acharya Narendra Dio College of Pharmacy. Rameshwar Singh of Acharya Narendra Dio College of Pharmacy. Harish Chand, again from Acharya Narendra Dio College of Pharmacy. Trisha Shukla, again from Acharya Narendra Dio College of Pharmacy. Dilbaz Ahmed from Mission College of Pharmacy. Tanu Pandey from Mission College of Pharmacy. Rajlakshmi Pandey from Bhagwat University, Ajmer. Dr. Anupam Sachin from Dayanand Dinanath College of Institute of Pharmacy. Dr. Piyush Yadav from Department of Pharmacy, Prasad Institute of Technology. Dr. Ganesh Lal from KJ College of Pharmacy, Varanasi. Dr. Mithilesh Dwivedi of KJ College of Pharmacy, Varanasi. Ms. Priya Dule of NES Gangamai College of Pharmacy, Dule. Satyaprakash Shukla of Hakikullah Chaudhary College of Pharmacy. Ajit Kumar of Abhishek Pharmacy College, UP. Ramkishore Kushwaha of Mankind Pharma. Urvashi of JSS College of Pharmacy, Jammu. Sushil Yadav from Prasad Institute of Technology. Ms. Neha Parveen from Prasad Institute of Technology. Abhay Kumar Mishra from Neelkant College of Pharmacy. Saloni Yadav from Acropolis Institute of Pharmaceutical Education and Research, Indore. Dheeraj Kumar Tiwari from Rajnarayan Pharmacy College, Jaunpur. Sachin Kumar Yadav from RDS College of Pharmacy, Jaunpur. Shreya Srivastava from RDS College of Pharmacy, Jaunpur. Ziba Fatima from SN College of Pharmacy. Shefali Agrahari from SN College of Pharmacy. Sapna Singh from SN College of Pharmacy. Sakshi Gupta from SN College of Pharmacy again. Maninder Kaur from GSRM Memorial College of Pharmacy. Jaya Pathak of Ashok Singh College of Pharmacy. Shivam Yadav of Babu Banarsi Das University. Tanu Sharma from Faculty of Pharmacy, Bhopal. Kushalendra Pratap Singh from AIMS.
It's now time for the most awaited prize distribution ceremony for various competitions that were held throughout the day. It's time to recognize and honor the exceptional talents and efforts of our participants. So let's gather around with excitement and anticipation as we celebrate the achievements of our winners. So first we have the winners of poster presentation. The first category is Pharmaceutical and the bronze medal goes to Sahil Srivastav. Next up, silver medal goes to Fahauddin. Finally, the gold medal goes to Purnima Sinha. The next category is chemistry and the bronze medal goes to Saloni Yadav. The silver medal goes to Priyanka Gupta. Okay, she might not be here. Priyanka Gupta, are you here? The gold medal goes to Amisha Yadav. Amisha. The gold medal goes to Amisha Gupta. The next category is pharmacology and the bronze medal goes to Shrishti Srivastav. medal goes to Suraj. Finally, the gold medal goes to Urvashi. The next category is pharmacognosy, and the bronze medal goes to Dr. Ganesh Lal. The silver medal goes to Devanshu Sahu. And the gold medal goes to Sulakshana Pal Singh. The next category is interdisciplinary. The bronze medal goes to Bharat Kumar. The silver medal goes to Aditya Rode. And the gold medal goes to Dr. Priya Jain. Now let's move on to the next competition, prize distribution, which is the quiz, the RX Quest, and the second runner-up is Ankur.
नेम रिपीट कर दें.
Second runner-up is Ankur.
सेकंड ठीक है सही है. The runner up. Next.
The first runner-up is Mahfouz. The winner of the RX Quest quiz is Dipali.
Fabrica model presentation.
The next competition was Fabrica model presentation, and the first runner-up is Team 2, which comprises of Puneet, Rohan, Tanisha, Nafisha and Shweta.
हाँ RKGIT गाजियाबाद.
RKGIT गाजियाबाद. And the winner for this competition is Team 1, comprising of Nandini Sharma, Vishal Gupta, Jitendra Bansal, Gunjan and Harsh Tyagi, RKGIT Ghaziabad.
सर बस आखिरी इवेंट है कैसे उसमें कितने दिन में 3 दिन अंदर मतलब नाम लेने एक साथ एक ही बार में नहीं टीम 1 टीम 2 टीम 3 अच्छा ठीक है हाँ
Finally, the last competition, which was the case study competition. The second runner-up is Team UIP, comprising of Shambhavi Tripathi, Akshita Srivastav, Virendra Tripathi and Sachin Shukla. The first drug dynamos, comprising of Rasha, Ankur Shetty, Manjeet and Komal. The winner for the case study competition is Team A1, Shubhojit Rai. Once again, a very hearty congratulations to all the winners. I would like to request Devansh to deliver the vote of thanks.
फोटो सेशंस के लिए
As we draw the curtains on this enlightening international conference on pharmaceutical innovations and spirit, the annual Technopharma Conclave, I, Devansh Jain, welcome you to the closing moments of what has been a truly remarkable journey. We have witnessed the convergence of brilliant minds, all united by a shared vision to innovate in healthcare and technology. On the behalf of the organizing committee, I extend a heartfelt thanks to all the speakers, delegates, and participants who have graced us with their invaluable insights. Your contributions have not only enriched this gathering but have also paved the way for future innovations in pharmaceutical industry. A special mention to our keynote speakers whose thought-provoking presentations have sparked a spirit of inquiry and passion within all of us. Our gratitude extends to our partners whose support has been instrumental in the seamless execution of the event. Special thanks to our HOD and the organizing chairman, Professor S. Hemlata ma'am, and the organizing secretary, Dr. Dinesh Rawat, and the co-organizing secretaries, Dr. Jairam Meena and Deepak sir, for the invaluable leadership and support in the helping us for the complexities of organizing such a remarkable event. Special thanks to my co-conveners, my colleagues, Abhirvay Iyer, Saurabh and Ayushi. Thank you for all the late and all-night discussions. Thank you to the whole team of Spirit. Thank you the anchors, the registration desk, the POCs and everyone who contributed. I'm filled with profound gratitude for all the staff and faculty of the department for their tireless commitment. Thank you one and all for making the International Conference on Pharmaceutical Innovations and Merit a memorable and one-of-a-kind experience. Thank you, everyone. Ma'am, I request you for a photo session. Ma'am, photo session. Yes. I request all the faculties of our department for a photo session.
गेस्ट स्पीकर गेस्ट ठीक है उसके बाद वॉलंटियर टीम का
ओके
उसके बाद पार्टिसिपेंट्स
I request all the guests, all the keynote speakers for a photo.
Volunteers, all volunteers, please.
Everyone, team spirit.
वो गया भगत आपसे मिलके ना क्या बोले यार तो हमको जाता है
Everyone, national anthem. Everyone.
पंजाब सिंध गुजरात